Assalamu alaikum. Cell surface extensions. In the previous lesson, we discussed the cytoskeleton. Now, we'll talk about the cell surface extensions that extend the cell membrane and project out from the cell surface. They have the same components as the cytoskeleton, with some differences. They are divided into four: Microvilli, Cilia, Flagella, and Stereocilia. These surface extensions have several different functions, such as cell movement, absorption, and dust disposal. Let's start with microvilli. They are finger-like cytoplasmic extensions whose main function is to increase the surface area to facilitate absorption. This is why it is mainly found in the cells that carry out absorption, the cells of the intestine, the proximal tubules of the kidney, and the cells of the gallbladder. 1 microvillus is 1 micrometer in length and 0.1 micrometers in diameter. They are found in large numbers, with approximately 3000 microvilli on the surface of the cell. It consists from the inside of actin filaments, numbering between 20 and 30, and its surface is just an elongation from the cell membrane itself. And if you remember what we mentioned in the cytoskeleton lesson, that the actin filaments are light and flexible, and from here we conclude that microvillus can be pulled and stretched and thus can increase the surface area of the cell. And this is exactly what the intestinal cells do during absorption. These actin filaments are connected by the helper proteins fibrin and villi. The microvilli contains enzymes that participate in the absorption process as well. We took information about the microvilli and proved how they participate in the absorption process. And now let's move on to cilia and flagella. They are cytoplasmic extensions, both of which consist of microtubules that make wave-like movements that help them move. And this may be for moving substances across a surface or moving the cell itself. Cilia are short, 5 to 10 micrometers in length and 0.2 micrometers in diameter. They make movements to move away liquids or dust on the surface, or they may move the cell itself in some types of microorganisms. They exist in large numbers, between 250 or more in a single cell. It is found mainly in the cells of the respiratory tract because it pushes back any dust toward the pharynx so it can be coughed up. It is also found in the oviduct of the female, where it pushes the egg with the same mechanism. As for the flagella, they are much longer than the cilia. Their length is 100 micrometers or more. And unlike cilia, the flagella are present in the cell in very few numbers. In most cells, it is only one. And since the flagellum is very long, its main function is to move the cell itself. The only example in the humans is the sperm in men, which moves using its flagellum, and there are many examples in bacteria. Cilia and flagella are both motile, and both have the same structure, the only difference being the length. Let's learn about their structure. Each of them consists of microtubules distributed as the following: 9 doublets of the 2 microtubules, A tubulin and B tubulin. And in the middle, there are 2 microtubule singlets, and we call this shape 9+2 axoneme. Many helper proteins are associated with these microtubules, the most important of which are nexin, which binds adjacent tubules, and dynein protein, or dynein arm, which is the protein responsible for moving them, and they cannot move without this important protein. The dynein protein uses ATP as an energy source to move the tubules. There is a special case found in the sperm, since it contains additional accessory structure that surrounds the axoneme. So it becomes 9+9+2 instead of 9+2 in the normal flagella. Dynein deficiency causes a genetic disease called Kartagener syndrome. In this disease, the cilia in the respiratory tract cannot move, and thus breathing becomes difficult for the patient. And the flagella in the sperm cannot move either, and thus the man carrying this disease becomes infertile. Both cilia and flagella have a strong basement in the cell, called the basal body. Its diameter is 0.2 micrometers, and its length is only 0.4 micrometers. Its structure is 9 triple bundles of microtubules. It does not have a specific function, it only acts as a base for cilia or flagella. After we learned about cilia and flagella, before we move on to the stereocilia, it must be mentioned that the cilia can move in one direction only. So they push the dust from the respiratory tract to the upside, and they push the egg from the ovarian canal towards the uterus in one direction. But the flagella can move freely, and thus the sperm or germs swim in the fluid using them. Now let's move on to the stereocilia, which are non-motile, unable to move. Scientists first called them stereocilia because they resembled cilia but are non-motile. But later they were able to discover them on the electron microscope, and they saw that their structure is very similar to the structure of microvilli, which are different from cilia, as we know that the microvilli are made of actin filaments, and the cilia are made of microtubules, so they changed their name to stereovilli, but most references still use the term stereocilia. The structure of stereocilia is similar to the one of microvilli, except that it is much longer and its actin filaments are irregular, and thus they are non-motile. But its function is also increasing the surface area to facilitate absorption, like microvilli, but in a weaker way. It is present in some places in the body, like epididymis. Now let's do a quick review. The cell surface extensions extend the cell membrane and project out from the cell surface. There are 4 types. Microvilli, which consist of actin filaments that can be bent. They increase the surface area, which facilitates absorption and digestion, and they do not require any energy. And since it helps with absorption, it is mainly present in the digestive tracts. Cilia and flagella consist of 9+2 shaped axoneme microtubules. The protein dynein moves them using ATP as an energy source. Both sit on a basal body. The cilia move the dust in the respiratory pathways and the egg in the ovary in one direction, and the flagella allow the sperm and the bacteria to swim in the liquid. Stereocilia are nothing like cilia. They rather look like microvilli, but they are non-motile and irregular. This concludes our discussion about the subject. If you want to test your knowledge by solving the questions, continue the video. The microvilli contain actin filaments, not microtubules. They have about 20 to 30 actin filaments inside them. Both cilia and flagella have a base in the cytoplasm, which is composed of 9 triplet bundles, while the 9+9+2 is a specific structure only in the flagella of the sperm. We said that the structure of the stereocilia is not similar to the cilia, but it is similar to the microvilli, so we also call them stereovilli.